Start. Okay, Bismillahirrahmanirrahim. Assalamualaikum. Okay, today's lesson is on nutrition and the human digestive system, as you can see in the slide. Okay, so this is on chapter 9, form 4. All right. Okay, now let's go to the second slide. Okay, the introduction for the second slide is on the digestive system, the one that we have been learning since form 2. Okay, it is made up of a long and a muscular alimentary canal. Okay, you must highlight the word muscular. Muscular means that the alimentary canal is a muscle that able to contract and relax. Okay, we will take a further look on that after this. Okay, and the alimentary canal starts from the mouth to the anus. Okay, let's see now where is the location of the alimentary canal. Okay, as you can see over here, based on the diagram. So the diagram showing to right now is where the alimentary canal starts from the mouth until the anus at the bottom, and then at the mouth and the alimentary canal is where the food will be chewed and move towards the stomach, towards the liver, towards the duodenum, and then the intestine. All right, move on to the next slide. Okay, there are two types of digestion as we can see in my slide, which is known as the chemical digestion and the physical digestion. And according to the textbook, let's go on to the first one, the physical digestion. Okay, this is where all the food is break down to form small particles. And in order to make that happen, we need two processes, which is the chewing, of course in the mouth, and the peristalsis, which is the movement of food along the alimentary canal. Okay, and then the next one is the chemical digestion. The word chemical itself will give you the clue that it will involve enzyme. Okay, and also this is where the small food being decomposed into molecule level and into simple molecules. Okay, so that is the basic thing we will learn from the physical food into the simpler molecule. And there is reason why the food need to be broke down into simpler molecule, because in order to be absorbed at the small intestine. Okay, so roughly we can say that before it's being absorbed, it cannot be absorbed in the big amount or big size. It must be absorbed. Before that, it must be turned into the smallest or the simplest molecule in order to be absorbed into the villus at the small intestine. Okay. Next slide. Okay. Again, this is the diagram of the digestive system. Okay, so the food will start the process of digestion in the mouth. So to make things easier, so we will only focus on three types of food, okay, which are the carbohydrate, protein, and lipid. Okay, so most of the food that goes into our mouth contain high amount of carbohydrates. So this is where the food will be started to be digested, or we also use the term hydrolyze. Okay, so carbohydrate will contain starch, and when the starch is being hydrolyzed, according to the question, is adding with water, and with the help of amylase enzyme from the saliva, it will break down into maltose, Hopefully all of you still remember what is maltose, which type of maltose carbohydrate belongs to. Okay, we have monosaccharide, disaccharide, and polysaccharide. So maltose belongs to the disaccharide, meaning that the carbohydrate or the starch, which is belong to the polysaccharide, starts to being simple into the next level, which is the disaccharide. And this is helped by the amylase enzyme. Okay, next. So once the food is being chewed, the food will be moving down to the esophagus with the help of process called peristalsis. Okay, as you can see the diagram over there, on the right side is the food that being turned into bolus. Okay, smaller particle from the original food. Thanks to the our teeth. Okay, so the food is chewed into smaller particle in the form of bolus, and from the bolus it will be moved down to the esophagus with the process of peristalsis. Okay, let's take a look. What is the meaning of peristalsis? Okay, according to the textbook, it is a rhythmic contraction and relaxation of muscles along the alimental canal. Okay, so the food will be moving along with the help of peristalsis and goes to the next destination, which is the stomach. So by using our understanding, the food is moving down not by the gravity. Okay, so if it happen I turn you upside down, so your food will still going to the stomach without any problem. Right, next slide. So we have done the first digestion in the mouth, which involve the carbohydrate. Okay, now let's go to the second place where the digestion will take place, which is in the stomach. Remember, just now I said we have 3 food class. So right now one have done, but it's not completely digested yet. Okay, the carbohydrate is now have digested into disaccharide. And now the food will move on to the next destination, the stomach. Okay, as you can see here, this is the cell wall of a stomach. We have cells that's being adapted to help the process of digestion. Okay, so in the stomach, as you can see, we have the surface mucus cell, we have the chief cell, and we have the parietal cell. Okay, these three cell is being stated in the textbook as the cell that form what we call the gastric gland. Okay, gastric gland. Gastric means the enzyme. Okay, the juices that help to digest the food. Okay, according to the next slide. Okay, so this is the cells that is being adapted to form the wall of the stomach: chief cell, mucus cell, and parietal cell. Okay, let's see what does each of these cell function as. Okay, the first one, let's take a look on the chief cell, right? The chief cell will secrete pepsinogen, which will, according to the name as well, the substrate will be protein and the enzyme will be pepsin, and that will turn the protein, the complex protein, into simpler, but not simplest, just simpler, which is into polypeptide. Okay, so in other words, we can say that at the stomach, the protein is not being completely digested yet. It just turned into a simpler molecule, which is the polypeptide. Okay, and the mucus cell, the function is to protect stomach. And lastly, the parietal cell. Okay, parietal cell will secrete the hydrochloric acid. Okay, what is the function of hydrochloric acid? Okay, there are three main functions. In lower form, we know that the acid is need to use to kill the bacteria. Maybe there is some bacteria have infected your food. So with the help of HCl or hydrochloric acid, it will help to kill the bacteria. Okay, and then it also act as inhibitor where the salivary amylase action that starts from the mouth is being stopped when it combining with HCl. Okay, and also HCl or hydrochloric acid also will prepare a medium for suitable pH for pepsin to react. Okay, so anything that goes less than acidic will disrupt the process of digestion. Okay, so right now let's take a look on the next location of digestion, which is happening at the small intestine. Okay, now the food is moving from the stomach. Okay, let's review back what just happened. We all know that at the mouth, the starch is now being digested into disaccharide, which is into maltose. Okay, the protein also have digested, which is now in the form of polypeptide. So only the lipid is still survive, not being digested. Okay, so now the movement of food is continuing from the stomach to the small intestine, and before they reach the small intestine, they will be going through a structure yang macam bengkok tu kan, okay, that is called the duodenum. Okay, and when they are traveling through the duodenum, they will crossing over the liver, where the liver will secrete the bile. Okay, kita tengok dekat sini. Oh, kat sini dah habis dah. Okay, kita tengok balik. Okay, so right now let's focus on the digestion of lipid. So when the food is moving through the duodenum, they will meet the liver, which will secrete the bile. Okay, bile doesn't digest the food. Okay, bile doesn't involve in digestion. What I mean is it doesn't break down, but the bile which is produced from the liver will emulsify the lipid into tiny droplets. Okay, kenapa kena tukar kepada tiny droplets? Kenapa kita tak biarkan saja? Okay, by turning the lipid into tiny droplets, we all knew that now the lipid will have higher surface area. Okay, so meaning that if we put or secrete the enzyme onto tiny droplets compared to the lipid itself, the reaction on the tiny droplets will be much faster. Okay, so that is the function of bile secreted by the liver. Okay, and then when the food will continue the journey, they will come over a structure called pancreas, where the pancreas will secrete enzyme, and that is where all the digestion will be completed. Okay, so before the food is reaching the pancreas, let's see now the latest condition of the food. Okay, let's start with carbohydrate. The carbohydrate is now in the form of disaccharide or maltose. The protein is in the form of polypeptide, and the lipid is now in the form of tiny droplet. All those three food classes is just halfly digested. Dia belum jadi lagi bentuk yang paling ringkas. Okay, or into the simplest form that can be absorbed at the small intestine. All right. So when it reaches the pancreas will finish up the digestion. Macam mana dia finish up? Okay, the pancreas or the duodenum will secrete pancreatic amylase. Okay, pancreatic amylase ni pun sebenarnya that is speciality for the carbohydrate that is that miss the digestion in the mouth. Okay, maksudnya kalau kita makan banyak karbohidrat, tak semua karbohidrat yang kita makan dalam mulut tu akan bercampur dengan air liur, or in other words, the saliva doesn't able to digest it or turn the food, all of the food into disaccharide. There are some of it is still being missed, but no worries. When the bolus reaches, when the food or the chyme reaches the duodenum, they will be digested by the pancreatic amylase into disaccharide. Okay, and then as for the polypeptide, kan tadi protein jadi polypeptide kan. Okay, so the polypeptide will be digested into peptides. Okay, peptides which is much simpler, polypeptide and peptide. Poly means banyak or panjang. Okay, peptide means single. Okay, so that is the action of enzyme trypsin. And lastly, the tiny droplets just now, lipid tiny droplets will be secreted by lipase and digested into fatty acid and glycerol. Okay, so all these enzyme is known as the intestinal juice. Okay, intestinal juice that is contain maltase, sucrase, lactase, lipase, and erepsin. Okay, and the surrounding of the enzyme of the duodenum is alkaline. So finally, before entering the long structure of small intestine, the carbohydrate is now as being digested into glucose, protein into amino acids, while the fat and the oil is now in the form of fatty acids and glycerol. Okay, so all of this now in the simplest form, it cannot be broke down anymore because this is the basic unit, or we call it monomer. Okay, so once it is in the form of monomer or the basic unit, it is ready to be absorbed at the small intestine. Okay, take a look at this slide. This is the view inside a small intestine. As you can see, it doesn't look very smooth because inside the small intestine we can see a lot of villus, okay, coming out from the wall of the small intestine. Okay, and the villus itself will provide a large surface area for absorption. Okay, let's take a look on the structure of the villus. Okay, so at the villus, we will focus on two structures that is responsible for the absorption of the digested food. Remember, we only have three simple molecules that are now moving through the villus, moving through the small intestine, which are the glucose, the amino acid, and also the fatty acid and glycerol. So these three digested food will now be absorbed by the villus, either in lacteal or in blood capillary. Okay, as you can see now, the blood capillary, which is in the blue, will absorb the glucose and amino acid, and the lacteal itself will be absorbing the fatty acid and glycerol. And since we have learned in Form 5 that lacteal actually is the extension or the tiny lymphatic vessel. Okay, so this is where all the fatty acid and glycerol will be flowing into the circulatory system. Okay, this diagram I took it from the textbook. That is showing what kind of digested food is being absorbed into the small intestine. Okay, so the method of absorption, you have already learned that in Form 4, Chapter 3, if I'm not mistaken, the movement of substance across the plasma membrane. Okay, so for example, let's say let's took on the fructose. Okay, fructose will be absorbed into the small intestine by facilitated diffusion, glucose by active transport, amino acid by active transport, vitamin B and C by water, and for water be absorbed by osmosis. Okay, and then the fatty acid and glycerol will be absorbed into the villus by simple diffusion. The same thing goes for vitamin A, D, and K. Okay, summary. So after absorption is assimilation that will be carried out by another speaker. So for my part right now, as you can see, let's take a look on the digestive system. We have the digestion, absorption, assimilation, and defecation. Okay, so let's review back what we have learned until absorption, meaning that the food is starting from the, sorry, I will take you back um, to the earlier slides, the component of the digestive system. Okay. Okay, so right now what we have learned is now the food. This is just a review again what I have just shared with you. So the digestion will start from the mouth, where the carbohydrate is being digested into disaccharide, into maltose, and then the food is being chewed by physical digestion and turned into bolus. So the bolus will move along the esophagus by the contraction of the wall of esophagus, the muscular wall, and then it will reaches the stomach. So in the stomach, the bolus will be changed into the new form of food that is called the chyme. Okay, and from the stomach, the next digestion occur is the protein, which is being digested into polypeptide. And finally, before all the food is being absorbed, the duodenum will secrete enzyme, or we call it the intestinal juice, where all the halfly digested food is being prepared into simpler molecule. So the end product of the carbohydrate will be glucose, protein will be amino acids, and fatty acid and the lipid will be turned or digested into fatty acids and glycerol, and they will move along the small intestine and to be absorbed by the villus. There are millions of villus inside our small intestine. Okay, so the villus will absorb all those three in different method at different places, where the fatty acid and glycerol will be absorbed at the lacteal, and amino acid and glucose will be absorbed at the blood capillary. Okay, so that is all from me. Okay, so we will continue our lesson with our next speaker, Cikgu Rahimah Razak. Thank you.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera. Terima kasih Cikgu Mio. Nampak tak slide saya?
Tak nampak.
Nampak.
Tak.
Okay, sekejap ya.
Tunggu.
Ada dah.
Okay. Akan sambung slot ni ya. System, chapter Assimilation ya. Okey, sebelum kita pergi lebih jauh ya, suka cikgu perkenalkan diri ya. Mungkin ada yang dah kenal ya. Nama cikgu Rahimah binti Razak ya. Sekarang mengajar di MRSM Merbuk ya. Sebelum ni pernah mengajar di MRSM Kuala Lipis, kemudiannya MRSM Serting, dan seterusnya Baling, dan insya-Allah sekarang sampai pension kot insya-Allah dekat Merbuk ya. Cikgu tua dah ya. Lagi 18 tahun nak pension ya insya-Allah. Okey, so cikgu tak nak cakap panjang ya. Apa yang cikgu nak kamu dapat ya, paling penting dalam masa 2 jam ni ya, untuk slot Nutrition and Human Digestive System hari ni, cikgu nak kamu rasa orang kata apa? Ceria, happy, seronok, enjoy saja. Ya, cikgu nak dalam satu minda kamu tu, kamu fikir yang bio ni sebenarnya satu subjek yang senang. Subjek yang senang, bukan subjek yang tough maksudnya bukan banyak benda mengingat. Cikgu tak nak kamu ada mindset macam tu lah ya. So kamu anggap bio ni senang Okey. So bio ni actually kita belajar tentang diri kita je kan ya. Tak kenal maka tak cinta kan. Siapa yang tengah bercinta tu kan. Make sure kenal diri sendiri, tahu dulu pasal diri sendiri dan sistem apa-apa lagi sistem proses dalam badan kita. And then baru boleh kenalkan nak cinta orang lain kan. Okey. So seterusnya cikgu nak sambung lah ya. Okey. Sebelum ni dengan Cikgu Mio tadi kamu dah belajar berkaitan dengan digestion process ya. And then sekarang cikgu digestion process dan juga absorption Satu perkara yang kamu perlu tahu, okey, anak-anak pelajar, makanan yang kita dah makan, makanan yang kita dah makan tu, dah masuk mulut and then melalui proses digestion, and then tak semua makanan ni dia adalah fully digested, not all fully digested. So kalau makanan yang kita makan tu fully digested, seterusnya dia akan melalui proses absorption, kemudiannya assimilation. Apa jadi dengan makanan yang tak fully digested? Contohnya macam fiber, water, yang tu kita akan tengok. Kalau undigested food, dia akan melalui proses diffusion. Tapi kalau fully digested food, contoh amino acid glucose, dia akan melalui proses assimilation. Kita nak tengok slide seterusnya, assimilation. What is the meaning of assimilation? Assimilation. Assimilation kalau bahasa Melayu dia disebut asimilasi. Actually assimilation is the process of transportation of digested nutrient. Digested nutrient that is simple compound. Simple compound tu apa dia? Glucose, amino acid yang diserap waktu proses absorption tadi. So into the body cell where they are used to form complex compound. Basically assimilation occurs in a body cell and also in a liver. Nampak kan? Ada gambar body cell, liver tengok GIF ni, GIF picture ni. GIF picture ni adalah ataupun gambar yang bergerak kan. Kamu tengok boleh nampak semua absorption and assimilation ni connection dua-dua proses ni. So kalau kita tengok dekat diagram ni, dekat GIF ni GIF picture ni. So dekat blood capillary kita boleh nampak ada kat sana amino acid monosaccharide, actually glucose lah dia akan absorb, dia akan absorb melalui blood capillary, dia akan absorb melalui blood capillary and then will be transport to the hepatic portal vein, into hepatic portal vein. Okey, kemudiannya kita tengok dalam lektin pula, ada lektin. Okey, lektin kita ada fatty acid ni sambung kat sini, tak nampak ni. Fatty acid, and then kamu nampak kat sini dekat L lektin, lemak. So cyclomicron, okay, cyclomicron actually combination of lipid molecule, for example fatty acid, glycerol, kolesterol, dia akan masuk ikut lektin, Kita tengok kat sini lektin it is connected to the lymph vessel. Okay, so tadi lipid semua tadi tu, okay, akan di bawah, okay, akan bergerak lah, okay, along the lymph vessel, and then it will move to the thoracic duct, okay, thoracic duct, and then pergi ke mana ni? Terus ke left subclavian vein, akan terus ke left subclavian vein, and then lepas tu dia akan sambung dekat, berjumpa dekat blood circulatory system lah, Okay, so kalau tadi cikgu ulang lagi sekali, untuk glukos dengan amino acid tadi dia transport, dua-dua bahan ni transport terus ke hepatic portal vein. Lepas hepatic portal vein dia akan terus pergi ke blood circulatory system, and then akan jumpa, kita akan hantar ke body cell lah, blood circulatory system. Okay, boleh faham Okay, so nak make sure kamu semua faham ni. Okay, lebih baik lagi cikgu akan tunjuk satu video.
Assimilation and the...
Okay, boleh dengar tak?
Boleh cikgu.
Boleh okay, terima kasih atas respon. Okay.
The liver produces and releases in the blood many useful substances. These substances are produced using absorbed nutrients. Digested nutrients are transported to the liver from the ileum by the hepatic portal vein. The liver is therefore able to regulate the amount of nutrients that reach the body cells. The liver does this by sometimes storing and sometimes processing the food it receives from the intestine and then releasing it at a rate which reflects the body's needs. The liver is involved in regulation of blood glucose. The liver, together with the pancreas, controls the blood glucose levels. It is very important that the blood glucose levels are very tightly regulated. Firstly, because glucose is the body's main energy source, and cells must be provided with the adequate amount they require, depending on the situation. And secondly, a slight change in the...
Okay, sikit je dia berkaitan dengan assimilation So cikgu harap kamu clear bab ni assimilation, digested food tadi tu dia akan transfer ke liver, okay, glucose dengan amino acid, manakala grup-grup lipid akan transfer ke thoracic duct and then terus ke left subclavian vein Okay. Okay, next slides. Okay, boleh nampak ke student?
Boleh.
Okay, alhamdulillah Kasih atas respon. Okay, next we move to function of liver. Okay, function of liver. Okay, the first one, okay, fungsi liver. Okay, so yang pertama sekali act as a regulator that controls the quantity of nutrient that enter blood circulatory system Okay, regulator ni apa? Okay, pengatur kawal atur Okay, and then metabolize, okay, digested food into substances, okay, that is assimilation kan. Apa dia digested food tu glucose, amino acid. So glucose ni it is used for cellular respiration. Amino acid it is used for synthesize plasma protein and enzyme Okay, now the third one, function of liver to expel toxic substances, that is urea from the blood, which is then it will be excreted through the urine Okay, and then last one, okay, kita tengok actually last but not least okay, kita tengok seterusnya function liver adalah act as a storage of excessive nutrient, okay, like glucose is converted into glycogen ya. Kalau glucose lebih-lebih dia akan di-convert, akan ditukarkan kepada bentuk glycogen ya. Okay, so kalau kamu tengok dekat diagram ni ada dua gambar kat sini, satu gambar normal liver satu lagi liver cirrhosis, okay, liver cirrhosis. So insya-Allah kita normal liver lah ya. Okay. Kalau orang yang liver dia, hati dia ni jenis yang cirrhosis ni, liver cirrhosis, okey, liver dia macam mana tahu? Asalnya liver dia normal, kemudiannya dia punya tisu-tisu liver yang normal tu dia dah bertukar menjadi skar ni apa bahasa Melayu? Parut kan, dia tumbuh parut macam tu kan. So bila dia jadi macam ni ya, actually dia punya fungsi normal liver tu dia tak berfungsi dengan baik lah. Okey. Bila letak fungsi yang baik, fungsi-fungsi yang keempat ni dia tak boleh jalankan lah ya. Okay, so actually semua ni berlaku disebabkan apa tahu? Kalau seorang tu dia jenis yang alkoholik, okay, minum minuman keras ya, and then mungkin dia pernah dijangkiti oleh penyakit hati yang kronik ya, contoh hepatitis B ya. Penyakit hepatitis ni adalah berkaitan dengan liver lah ya, ada problem dekat ataupun infection dekat liver Okay, next we move to the next slide. Okay, that is assimilation in the liver. Kita tengok tadi dua bahan, okay, amino acid dan juga glucose kan. Okay, dua bahan amino acid dan glucose. Okay, first sekali amino acid ni kenapa dia nak pergi liver kan? Okay, apa yang berlaku dekat liver dekat liver amino acid ni akan disintesis into plasma protein dan juga enzim ya. Untuk apa? It is used for body cell function, that is for cellular respiration lah ya, plasma protein, enzyme, and also glucose juga akan digunakan untuk body cell function lah, for cellular respiration. Actually when the glucose supply is insufficient, okay, amino acid ni akan tukar kepada glucose lah dalam dekat kita punya body cell Okay, and then bila mana amino acid ni pula, kalau dia lebih-lebih dia lebih dekat kita punya kita konten dalam badan kita lebih dekat liver, amino acid dia akan undergo proses nama dia deamination. Dalam bahasa Melayu dia penyahminaan. Ya ke? Kita nak remove lah amino acid itu, kita nak convert kita nak broken down kepada urea. Kalau lebih-lebih amino acid tak bagus okay, kita akan convert kepada urea and then kita akan excrete through the kidney through kidney So actually kalau amino acid lebih dalam badan kita, dia akan menyebabkan kumpulnya uric acid, urea kan, dia boleh menyebabkan penyakit gout Okay. So that's why kalau orang penyakit gout dia tak boleh makan benda yang banyak protein seafood semua tu tak boleh makan, ikan bilis semua kan, macam tu. Alright, next glucose. Glucose buat apa pula dekat liver? Okay. So glucose dia akan used for for cellular respiration to gain energy, and it is used in the body cell Okay, bila mana glucose lebih-lebih pula dekat kita punya body cell tu dia akan jadi so glucose dia akan convert into glycogen, okay, and then dia akan store in the liver and the muscle and then apa jadi lagi kalau glucose ni dalam keadaan lebih lagi okay, dah tukar kepada glycogen, lebih lagi glucose tersebut dia akan convert pula kepada fat, okay, yang mana dia akan store dekat organ and underneath skin underneath skin dekat bawah kulit and then bila mana kita punya glucose dalam badan kurang pula, apa jadi? Okay, glycogen yang tadi tu ditukar glucose tukar glycogen ni pula akan ditukar balik kepada glucose macam tu. So fungsi liver ni sangat penting lah Okay. So next tengok pula apa yang berlaku dekat sel assimilation in the cell. So dekat sel okay, melibatkan tiga bahan Okay, tadi amino acid, glucose, lipid. Kalau dekat liver amino acid dengan glucose saja kan. Okay, kalau dekat sel ada tiga benda ni amino acid, glucose dengan lipid So okay, when amino acid akan disynthesize dekat body cell, amino acid akan disynthesize into protoplasm and protoplasm ni apa? okay, combination of nucleus, cytoplasm, protoplasm, plasm, cytoplasm combination kan. Okay, nucleus, cytoplasm and other organelle. Okay, and also amino acid ni juga dia akan disintesis untuk apa? To repair and build new tissues. Okay, repair damaged tissues. Okay, also to synthesize hormone and enzyme. Okay, all this, okay, it is used for the body cell function. Yeah. Okay, and then next kita tengok pula glucose. Okay, dekat blood circulatory system buat apa? Okay, glucose dia akan oxidize through the cellular respiration to release energy, which then it is used in the body cell. Bila mana glucose tadi dia dah lebih in excess, okay, dia akan convert kepada glycogen, sama juga tadi akan store dekat muscle. Okay, tapi dekat sel ada satu lagi, okay, bahan iaitu grup-grup lipid lah, okay, glycerol, fatty acid, vitamin A, D, E, K. Kat sini dia akan lipid tadi akan sintesis, okay, synthesis paling penting kamu eja betul, okay, into plasma membrane, okay, to form, cikgu tak pernah nampak kat sini, to fat tadi tu, plasma protein, okay, sorry, plasma protein akan digunakan apa tu? Okay, fosfolipid, plasma protein kamu tahu lah, plasma protein, astagfirullahalazim, plasma membrane, itu tak datang tu kan, okay, sorry. Okay, contoh plasma membrane apa dia? Dia punya fosfolipid dan juga kolesterol kan yang membentuk plasma membrane tu kan ada fosfolipid, kolesterol kan. Okay, so next apa ni lagi satu, dalam keadaan yang lipid tadi berlebih-lebih, okay, dia akan tukar kepada fat, ditukar kepada fat, and then it will keep, okay, all this fat are keep and then store in the store as a energy in the adipose tissue that is underneath skin lah, dekat bawah kulit, and also apa ni oxidation of lipid juga, okay, to release energy, okay, tapi dalam keadaan glukos yang tak mencukupi, okay, apa ni dalam keadaan glukos yang tak mencukupi, dia akan convert baliklah fat tadi kepada glukos, and then it is used for use in the body cell. Okay, cepat sangat ke cikgu ni, harap kamu faham, okay. Next, ini adalah summary, summary assimilation in the liver and cell, apa yang berlaku dekat dalam liver dan juga cell, So kamu nampak kat sini, okay, okay, glucose, amino acid tadi, okay, dia akan pergi ke liver dulu kan, lakukan proses-proses fungsi dia apa sekian-sekian, okay, tapi kalau kita tengok kat sini lipid, fatty acid, glycerol, dia akan terus pergi ke blood circulatory system. Okay, so ringkas cikgu boleh cakap macam ni lah, soalannya macam kalau lah ada dua family kan, dua kereta nak pergi bercuti dekat mana, okay, kat PD kan, okay, grup glucose, amino acid, okay, akan singgah ke KL, cikgu daripada Kedah, okay, manakala lagi satu kereta akan terus pergi ke PD kan, so macam tu lebih kurang, akhirnya nanti semuanya akan jumpa balik dekat blood circulatory system, okay, kamu tengok tadi. Okay, next. Okay, cikgu nak tunjuk satu contoh. Okay, soalan structure berkaitan dengan assimilation, combination, assimilation and absorption, Okay, cikgu bantulah sebab kamu tak boleh jawab kan. Kalau kamu boleh jawab ni, cikgu dah suruh minta jawab ni, Okay, so first kamu tengok diagram ni, diagram yang pertama, ditunjukkan proses act, that take place in the human ileum. Okay, manakala diagram tu shows digested food is carried from the ileum to the liver and the body cell. From the ileum, liver to the body cell. Okay, what is the process X? Okay, process X kamu nampak dekat mana ni? Dekat ileum. Okay, itu yang pertama. Apa proses X ni? Okay, yang kedua soalan dia, how does glucose? Glucose dari mana glucose? Okay, glucose kan. How does glucose diffuse into the bilis, macam mana dia boleh masuk ke dalam glucose tadi tu boleh masuk dalam bilis kan. Okay, pergi ke blood capillary ke, pergi ke lactil ke kan, dia minta explain, Okay, so cikgu terus tunjuk jawapan, Okay, kalau kamu tengok kat sini, proses X itu adalah proses absorption, proses absorption. Okey, siapa jawab absorption? Okey, betul Okey, yang kedua glukos, kalau yang B ni. Okey, kamu boleh nampak jawapan B tu. Okey, how does glucose diffuse in the villus? Ada dua markah kat sana, Glucose absorb into the blood capillaries through the epithelial cell. Okey, secara apa tu? Proses apa tu? By active transport. By active transport against the glucose concentration gradient, Okey, so simple kan? Semua orang boleh jawab, alhamdulillah. Okey, seterusnya satu lagi soalan, sambungan daripada soalan tadi, dia tanya adaptation, give one adaptation of villus to help process egg in the ileum. Okey, bagi satu karakter villus. Tadi Cikgu Mio dah explain kan. Kamu pilih mana-mana satu. Banyak tadi villus, karakter dia punya karakter dia. Okey. Ni antaranya one cell teat have a layer of epithelium, one cell teat got blood cells, secrete mucus, have a lot of blood capillary network, contain lactate, scan bla 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 kan. Banyak kamu pilih mana-mana satu sahaja cukup. Okay, next. Okay, salah satu lagi soalan. State the name of the process that take place in diagram 1B. Apa nama proses ni? Cikgu tunjuklah pula kan. Okay, ini proses apa ni? Ini adalah proses assimilation. Okay. Okay, next. Okay, so seterusnya kita masuk satu lagi subtopik berkaitan dengan defecation. Defecation. Kalau bahasa Melayu ni kita translate sebagai penyah tinjaan. Kalau in English, process of removing feces expel out of our body. Ingat lagi meaning of defecation. Bila mana kamu form 2 kamu dah belajar kan topik ini. Okay. So kita nak tengok defecation function of the large intestine. So kita tengok sikit. Okay, yang pertama sekali function dia, okay, absorption of water and vitamin. Vitamin apa? Okay, vitamin B, vitamin K and folic acid. Okay, kamu kena ingat tadi amino acid, glucose tadi tu, grup-grup lipid tadi tu yang digest, diserap. Okay, kemudian dia akan melalui proses assimilation. Apa jadi dengan bahan yang tak diserap? Macam contoh cikgu bagi tahu tadi, fiber, fiber and then apa lagi? Water, semua tu kan. So tak diserap, dia akan melalui proses defecation. Okay, inilah function defecation. Function large intestine absorption of vitamin. Okay, vitamin B, K tadi. Okay, lagi satu function dia adalah tu as a format of feces, formation of feces untuk pembentukan najis, pembentukan najis. Okay. Okay, after water is reabsorbed, the remaining waste is called feces. Feces contains dead cells by pigments, bacteria and toxic substance. So banyaklah benda dalam feces tu. So baguslah kalau kita boleh buang satu hari sekali ataupun 2 hari sekali. Kalau lebih daripada tu, okay, tak baguslah tak bagus. Some tips, satu tips saya cikgu nak share dengan kamu. Kalau kamu jenis yang susah buang air besar okay, nanti akan lead to the constipation. Cuba amalkan makan buah betik. Buah betik ataupun buah naga. Naga mahal sikit kan, buah betik murah sikit. Okay, insyaAllah kamu akan mudahlah nak buang hari-hari Okay, so bila buang hari-hari insyaAllah akan orang kata apa? Akan turunlah sikit. Hopefully turun sikit berat badan Okay, and then satu lagi function apa ni large intestine, that is apa tu? Wall of large intestine secrete mucus to smoothen the movement of the feces. Okay. Yang ni tak apalah, cikgu nak tunjuk satu video. Okay. Satu video ni panjang tapi cikgu akan ringkaskan berkaitan dengan defecation. Okay, start kat sini.
The small intestine is connected to the large intestine via the Bauhinsve.
Boleh nampak ke?
Boleh.
Boleh cikgu.
It opens when chyme is to pass from the small intestine to the
Okey.
large
Ini
intestine.
akan cerita pasal large intestine
The
ya.
large
Relate dengan
intestine does
defecation.
not have any villi like the small intestine because most digestible substances have already been
Ini
absorbed in the
bakteria
small
banyak
intestine.
dalam large
However,
intestine.
the large intestine has an estimated 100 billion
100
bacteria
billion.
inside. They are important for many other tasks, such as the production of vitamins and the decomposition of fiber for the body's energy production. Many of these bacteria are an important part of the immune system by killing harmful germs.
Okey, ini adalah
Through
pembentukan feces
peristalsis, the chyme is transported from the ascending colon to the transverse colon to the descending colon. On its way through the large intestine, water is removed from the chyme. Furthermore, mucus is added for proper excretion of waste. Substances that cannot be absorbed through the small intestine or the large intestine remain in the rectum and are finally excreted through the anus.
Okey, so habis akan dibuang lah, najis tu akan buang melalui anus ya. Okey. Ini tadi dah. Okay, next kita move to next slide ya. Okay. Okay, next kita tengok pula balanced diet. Okay, balanced diet is a diet that consists of all 7 classes of food in a correct proportion and balanced quantity according to the individual needs to maintain optimal health. Jadi balanced diet ni kita tak boleh cakap tiap orang tu sama, tiap orang tu dia berbeza. Tengok jugalah dia ni contohnya macam orang kata apa, kalau dia seorang yang perempuan yang sedang mengandung ke, remaja ke kan, dewasa, kerja kat mana kan, tengah panas ke, kerja aircon ke, semua actually balance diet berbeza untuk setiap orang ya. Okay, so kalau tengok daripada bawah ni carbohydrate yang ni cikgu saya tak payah cerita detail ya. Okay, cikgu nak tunjuk satu contoh berkaitan dengan soalan berkaitan dengan balance diet ni ya. Okay, objective question ya. Okay, kamu tengok gambar rajah ni, okay, dia ada bagi dua diagram. Satu diagram kamu nampak ni ada bread kan, ada cereal, so ini adalah carbohydrate lah. Okay, yang lagi satu ada chicken, ada fish meat, dia semestinya daripada group protein kan. Okay, soalan dia tak tanya apa dia group, apa dia X and Y. Type of food classes dia tak tanya, dia tanya selebih daripada lain daripada tu So kamu kena faham dulu X and Y, then baru kamu boleh jawab soalan ni. Which of the following health problem are caused by taking too much food in a long time? Apa jadi kalau kita ambil karbohidrat lama-lama? Okey, berulang-ulang untuk jangka masa yang lama, protein pun untuk jangka masa yang lama. Apa efek dia? Ya. Okey, so kamu rasa apa jawapan dia? Okey, untuk jimatkan masa cikgu bantu kamu Okey. So jawapan dia adalah okey, betul. C kan? Okey, so jawapan dia C So diabetes mellitus untuk X dia akan menyebabkan kalau diabetes penyakit kecil manis So kita tahu karbohidrat mengandungi starch dia akan convert jadi apa? Glukos kan? Macam tu. Jadi kalau glukos banyak badan kita, akan menyebabkan penyakit kencing manis lah. Ini tadi protein kalau amino acid banyak dalam badan kita uric acid berkumpul urea banyak dia akan menyebabkan penyakit gout Okay, next we move to diet modification for specific individuals Okay, ini yang dicadangkan Okay, makanan dicadangkan oleh Ministry of Health Okay, Pinggan Sihat Malaysia, namakan Pinggan Sihat Malaysia is a guide given by a Ministry of Health to promote good eating habit. Kita sepatutnya amalkan makanan ni, kita panggil apa? Quarter half ataupun suku-suku separuh. Suku-suku separuh, ambil starch ataupun nasi separuh, lauk separuh ke ikan ke daging ke ayam, and then yang selebihnya tu kita kena ambil fruit and vegetables yang mana group dia dalam fiber group fiber ni. Okey. Tengok satu lagi soalan, simple je soalan ni, jam mata boleh jawab kan. Okey, daripada yang tadi tu, gambar rajah yang tadi, okay, which of the following food class should to be taken most? Mana kena ambil paling banyak? Okey, jawapan dalam diagram ni juga. Okey, kamu nampak kan, yang ni suku separuh kan paling banyak ambil buah-buahan sayur-sayuran kan. Ni separuh kan sebelah, so jawapan dia adalah A, fiber. Okey, senang. Okay, next satu lagi soalan. Okay, cikgu tunjuk jawapan, sorry. Okay, soalan gambar raja yang sama. Explain why we should take only quarter portion of rice in our daily diet. Kenapa kita nak kena ambil nasi ni separuh je, suku je, suku bukan separuh, suku suku. Okay, kenapa tu? Kan? Okay, cikgu tunggu jawapan lah ya. Okay, first kali tadi relate dengan soalan yang tadi ya. Okay, to avoid ataupun to prevent from diabetes mellitus. It is because of rice contain starch. Rice contain starch. Okay, starch ni akan apa? Okay, starch is polysaccharide ataupun then dia akan convert into glucose. Okay, large number of glucose will lead to the diabetes mellitus. Ya. Okay. Next. Okay, health issues related to the digestive system and eating habits. Okay, yang ni pun penting juga sebab cikgu tengok banyak soalan pasien ni banyak berkaitan dengan soalan ni ya. Okay, gastric bypass procedure. Gastric bypass procedure. Okay, gastric bypass procedure actually bahasa Melayu dia bypass ni shortcut lah kan. Okay, kalau kamu tengok gambar rajah ni, gastric bypass involve reduction of stomach size by surgery. Buang sikit bahagian perut tu ya. Okay, reduction of stomach size. Kecilkan sikit saiz perut melalui kaedah pembedahan This procedure is usually done to individual with obesity. Problem ni kalau seorang tu dia gemuk sangat, badan dia gemuk sangat tapi and then dia nak kuruskan dengan jangka masa yang singkat. Dia boleh guna kaedah ni lah. Tapi hati-hati, kamu tengok dia ada short term side effect. Short term side effect apa tu? Dia ada side effect lah kan. Kalau short term side effect, lepas buat surgery ni, pembedahan ni, boleh menyebabkan acid reflux. Acid reflux ni soalan yang macam bahasa Melayu ni kita panggil heartburn. Pedih ulu hati. Kamu pernah tengok tak iklan Gaviscon kan? Dia makan Gaviscon minum Gaviscon tu, ubat Gaviscon supaya untuk mengelakkan heartburn pedih ulu hati. Kalau makan cepat-cepat pun boleh menyebabkan acid reflux. Acid reflux ni maksudnya apa tu, acid tak digest betul-betul Jadi acid tu dia akan naik sampai ke kita punya esophagus. That's why dia menyebabkan orang kata apa ni lah yang kita namakan sebagai acid reflux, nausea, nausea, loya, pening, berminting, muntah, expanded esophagus and also infection Expanded esophagus ni kita punya esophagus mengembang long term, side effect dia dizziness, low blood sugar level, malnutrition, stomach ulcer and also defecation problem. So cikgu nak explain sikit berkaitan dengan gastric bypass procedure ni. Gastric bypass procedure ni before ni normal kita punya perut, duodenum, esophagus, duodenum, small intestine. Sebelum pembedahan normal. Selepas pembedahan kita tengok ini dekat dia punya perut tu dia akan bentuk kantung kecil dekat perut ni, dia akan potong sikit, dia akan bentuk kantung kecil di perut, kemudian dia akan sambung ke usus kecil small intestine. Dia buat shortcut. So makanan yang masuk ikut sini lebih sikit lah bila makanan masuk dekat perut tu sikit, dia absorb sikit lah, makanan dia absorb sikit So dengan cara tu kalori makanan sikit menyebabkan dia makan sikit kurus lah macam tu. Long term tadi cikgu dah explain dizziness, pening, loya, low blood sugar level, malnutrition, dan sebagainya lah tengok sikit soalan ni. Nak habis dah. Example 5, structure, contoh yang kelima, satu lagi soalan structure. Apa ni? Doctor have done bariatric surgery as shown in diagram 2.2 to overcome the obesity problem. Ni samalah juga dengan bypass tadi yang cikgu bagi tahu tadi kan, gastric bypass surgery dia. Explain how the method can overcome obesity. Macam mana? Method kaedah bariatric surgery ni nak atasi obes cikgu tu explain. Jawapan dia adalah to reduce size of stomach, Adik buat ni dekat perut ni, dia potong sikit bahagian perut tu, dia sambung dengan small intestine. Small intestine ke apa kan, penting kamu eja betul Reduce size of stomach, okay. Then it will reduce total calorie intake. Bila mana perut kita makin kecil, makanan yang diserap pun sikit, So more body fat change to the energy, okay. So with that body weight akan decrease, Okay. Dapat tak yang tadi tu? Cik harap dapat. Nanti kamu boleh tengok juga past question, banyak past question berkaitan soalan ni. Nanti kamu make sure tengok balik Okay, kalau cikgu cepat lah Okay, next we move to health issues related to defecation. Health issues related to defecation. Penyakit-penyakit yang berkaitan dengan defecation penyah tinjaan ni. Okay, apa dia? Constipation Kalau seorang tu dia susah buang air besar, 3 hari, 4 hari tak buang-buang kan, dia kita namakan sebagai constipation, sembelit. Kalau constipation ni berulang lama-lama, dia akan menyebabkan hemorrhoid, buasir. Buasir ni biasanya berlaku pada lelaki lah perempuan boleh berlaku juga, tapi selalunya kalau perempuan berlaku bila mana lepas dia bersalin tu Okay, push kuat-kuat kan, yang dia boleh keluar buasir lah Okay, and then kalau long term lagi, effect lagi, dia boleh menyebabkan colon cancer dan juga rectal cancer Okay, so sebenarnya kita jangan biarkan mula constipation jangan mulakan yang constipation tu. Kita kena amalkan gaya hidup sihat, makan makanan yang disuggest tadi kan, suku-suku separuh untuk selamat untuk dapat kesihatan lebih baik jangan makan lebih-lebih Okay, how to prevent this health problem related to defecation? Increase the intake of fiber and water in daily diet. Kita kena banyak makan fiber, minum air banyak. Fiber tu macam cikgu cakap tadi kan, buah-buahan, sayur-sayuran itulah Okay, next. Okay, apa dia function fiber ni? Banyaklah function fiber Okay, stimulate peristalsis, absorb and expel toxic substances, regulate the absorption of glucose, especially for diabetes mellitus patient, and also to increase the population of good bacteria in the large intestine. Bakteria pun dalam badan kita ada yang bakteria baik, bakteria yang tak baik kan. So kita nak yang bakteria baik tu, so kena banyak ambil fiber Juga function of water, semestinya to soften kita punya feces untuk lembutkan kita punya najis, and also with that bila air kita minum cukup najis akan mudah keluarlah melalui large intestine. Okay, alright, last, health issues related to eating habit. Okay, boleh menyebabkan penyakit gastritis. Okay, eja bagi betul anorexia nervosa, bulimia nervosa, and also muscle dysmorphia. Yang ni kamu baca sendiri lah ada lantai sebut sama je, cikgu ambil yang ni Okay, so itu saja masa yang kita ada Okay, sebelum cikgu akhir sesi cikgu ni, suka cikgu ingatkan kamu tanya diri kamu setiap hari. Okay, jika kamu tidak berusaha untuk berjaya, jika kamu tak berusaha untuk berjaya, sebenarnya kamu telah berusaha untuk gagal. Sebagai seorang student, kamu tak ada pilihan. Kamu kena buat sesuatu setiap hari. Kamu kena buat sesuatu. Contoh macam hari ni kan, kamu dah join webinar Bayu kan. Sebenarnya kamu telah selangkah mencapai kejayaan. Kalau setiap hari tu kamu tak buat apa-apa, sebenarnya tanpa kamu sedari, kamu telah berusaha untuk gagal. Okay, itu saja daripada cikgu. Terima kasih yang join hari ini. Sekian, wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh. Seterusnya cikgu serahkan kepada cikgu Nukiman.
Boleh cikgu.
Okay, from MRSM Beto. So I will continue this 9.2. So if you have textbook, please open textbook. Okay, kejap. Dah share screen ke ni?
Belum cikgu Dikiman.
Belum lagi
Belum cikgu.
Okay. From Malaysia, in Malaysian, okay, daripada Sabah, Sarawak kan. Hopefully adalah daripada MRSM Betung tu sendiri datang kelas pada hari ini. So from my part, Okay, I continue because I only have 40 minutes to discuss my topic. So for my topic, I will discuss 3 experiments. Okay, still in chapter 9, that is nutrition and human digestive system. Okay, first experiment, activity 9.1, that is studying the digestion of starch in a food sample. Okay, 9.2, studying digestion of protein in a food sample, and last activity 9.3, studying the digestion of lipids in a food sample. The end of my session, you will play one game. We have a quiz, okay, and you will play one game. Okay, so I want you, all of you, focus in my session, my lesson today lah. Okay, so let's start with first experiment. Okay, activity 9.1. Okay, if you have textbook, you can open page 147. Okay, today I'm not teach you about the technique answering question from paper 3 because previous teacher already teach you the technique how to answer a question paper 3, right? Okay, today my focus is only share to you how should you handle experiment. Okay, how you should handle experiment because in paper 3, KSSM, new format, you need to handle experiment. You need to handle experiment. Okay, it's not same like before. Before paper 3, you are given the data and then you just write the data in the box, right? But for SPM, KSSM format, you need to conduct one experiment and the best is you must get accurate result. Kamu mesti mendapatkan keputusan yang tepat, okay, supaya selari dengan teori kepada eksperimen tersebut. Supaya selari dengan teori. Sebab kalau kamu salah buat eksperimen, of course the result akan salah juga. Okay, and then it will affect when you answer the question. Ia akan menjejaskan kamu ketika kamu nak menulis jawapan. Okay, terutamanya nantilah kamu tengok sendiri eksperimen balik maktab nanti, kamu bincanglah dengan cikgu kamu, so perbanyakkanlah aktiviti makmal. Okay, so before we start experiment, you should know about the background of the experiment. Okay, for activity 9.1, studying the digestion of starch in a food sample. Okay, Cikgu Mia already teach you about the digestion that occur in your mouth. In your mouth, okay, starch will hydrolyze, okay, with help with salivary amylase to produce maltose. Okay, here maltose is reducing sugar. Okay, maltose is reducing sugar. So in this experiment, we will use Benedict test to test the presence of reducing sugar. Okay, dekat tangan cikgu sekarang ada sandwich. Takut-takut lapar tengah hari ni kan. Kalau lambat makan tengah hari boleh kena gastrik kan. So this sandwich, dia ada apa? So it contain starch. So when I eat sandwich, first digestion occur in my mouth is digestion of starch. Okay, so ni gambar baby, ada air liur. So food, when it mix saliva, it will produce bolus and make it easy to transfer to your stomach, okay, through the esophagus. Okay, now we go to the experiment. Okay, so let's watching this video. Okay.
Okay, Assalamualaikum warahmatullahi wabarakatuh and hi everyone. So today
boleh dengar ke suara dia?
Study the digestion of starch in the food sample.
Before running an experiment, you must read the instructions that have been given. You are asked to identify problem statements, hypotheses, list of variables. Please check one by one the materials and apparatus used in this experiment.
Okay, so tadi sedikit tips daripada cikgu kalau kamu menduduki ujian amali sama ada SPM LSM ataupun SPM akan datang, first thing yang perlu kamu buat adalah baca dahulu arahan pada soalan. Baca dahulu satu persatu, kenal pasti problem statement, hypothesis, variable kan. Barulah kamu check dulu material apparatus, baca step, dia punya prosedur, barulah kamu menjalankan eksperimen. Jangan kamu bila pengawas cakap, okay, pelajar boleh mula, terus jalankan eksperimen. Elakkan. Okay, so let's continue the video.
Then, please run the experiment according to the given procedure. Please do the experiment step by step. For this experiment, student can refer page 147 in the biology textbook form 4. Let's start the experiment. First, label two test tubes A and B by sticky notes. Then, add 3 ml of 1% starch suspension to each test tube. Fill test tube A with 3 ml of 0.5% amylase solution and test tube B with 3 ml of distilled water. Soak both test tubes in a water bath with a temperature of 37 degrees Celsius for 10 minutes. After 10 minutes, remove 2 ml of the solution from test tube A . And put it into a different test tube. Add 3 drops of Benedict solution to that test tube. Heat the test tube in a boiling water bath for 1 minute. Record the color of the content. Add 2 drops of iodine solution to the remainder of test tube A. Repeat steps 5 and 6 for test tube B.
From the iodine test, from the test tube A, the color of iodine is remain yellowish color, while for the test tube B, okay, this is the test tube B, so the color of iodine is changed to dark blue color. Okay. And as per the Benedict test, so the test tube A, the color changes to brick red precipitate, and also for the test tube B, the color remain unchanged.
Okay, so for discussion, as you can see class, as you can see at test tube A, the solution in test tube A, when you put iodine, the color become yellowish. Okay, the color become yellowish because starch is not present in test tube A. Okay, so why starch is not present? Because all starch are broken down by amylase to maltose. Okay, so that's why starch is not detected when you when you test it by using iodine test. For test tube B, for solution in test tube B, when you put the iodine test, okay, the color of solution become dark blue. Okay, dark blue. This is because it detect the presence of starch. Okay, starch is not broken down into maltose because in solution B there are no amylase. Okay, there are no amylase in the solution B. So that's why in iodine test it detect the presence of starch. While for Benedict test, the solution, okay, in test tube A, the color change from bluish. Okay, the bluish color is from Benedict solution. Okay, bluish turn to brick red precipitate. When you answer the question, when you write your observation, you must write brick red precipitate. Okay, you must write brick red precipitate. Okay, so the color change because it detect the presence of reducing sugar. Reducing sugar in this experiment is maltose. Okay, so all starch are converted to reducing sugar by amylase. For solution B, okay, solution B, when you test it by using Benedict solution, the color of Benedict solution is remain same, bluish. Okay, because it not detect reducing sugar. Okay, because in solution B, there are no amylase to break down starch. So that's why the color is remain same. Okay, so conclusion for this experiment. Okay, if you read the hypothesis for this experiment is amylase hydrolyze starch to reducing sugar. So for conclusion, hypothesis is accepted. Okay, enzyme amylase converts starch to maltose that is reducing sugar, and the presence of reducing sugar it can detected by using Benedict test, where Benedict solution it turn to brick red precipitate. Okay, so we are finished for activity 9.1. Now we go to activity 9.2, studying the digestion of protein in food sample. Okay, background of this experiment is digestion that occurred in your stomach. In your stomach, okay, in your stomach, digestion that occur is digestion of protein. Okay, so if you see this chicken, tengah hari kan, tengok ayam panggang, perut pun dah rasa lapar kan, rasa trigger nak makan. Okay, so this chicken, it contain protein, rich with protein. Okay, this chicken rich with protein and it start to digest chemically in your stomach. Okay, so as Cikgu Mio said before, okay, your stomach, the wall of stomach, okay, it produce and secrete gastric juice. Gastric juice here, it contain hydrochloric acid that prepare acidic medium for activity of pepsin. Okay, hydrochloric acid, pH dia adalah pH 2. Okay, then it also contain inactive enzyme that is pepsinogen. Pepsinogen is active ataupun convert to pepsin when it react with hydrochloric acid. And this pepsin, it will react on protein. It will digest protein to become polypeptide. Mucus that secrete in the wall of the stomach, okay, it protect your stomach from corrode, okay, from corrosion. because of hydrochloric acid, and this renin for coagulate milk. So today I'm just focused for this part. I'm just focused on digestion of protein, okay, by enzyme peptine, and it will produce polypeptide. So we prove it by this experiment. Barulah kita jalankan eksperimen setelah kita tahu latar belakang kepada eksperimen tersebut. Okay, so let's watching this video.
Okay, now we move to the activity 9.2 to study the digestion of protein in a food sample. So let's start our experiment.
As usual, before we begin this experiment, we first list the following. First, the problem statement for this experiment. What is the effect of pepsin on protein? Second, for the hypothesis, pepsin hydrolyzed protein in the albumin suspension into polypeptides. And for variable, the list of variable is as follows. Manipulative variable, present of pepsin, responding variables, clarity or turbidity of mixture after 20 minutes, and fixed variable, temperature at 37 degrees Celsius, concentration of pepsin solution, and dilute hydrochloric acid. Again, be reminded that before you run experiment, make sure that the materials and apparatus needed in this experiment are sufficient. You need to run this experiment step by step. Let's watch how this experiment was conducted by teacher Wan Nurul Izzati. Let's watching the video.
for experiment 9.2, the result for this experiment, for test tube A, the changes of color from cloudy to clear, and also for test tube B, the solution remain unchanged, which is cloudy. Okay.
So for discussion class, okay. For test tube A, as you can see that the solution become clear. Okay, this is because pepsin that present in solution A, it breaks down albumin, okay, into polypeptide. Albumin here, okay, albumin daripada telur. So albumin adalah protein, okay, yang boleh dijumpai dalam telur. Okay, then hydrochloric acid, okay, that we mix in this solution, it provides acidic pH medium for pepsin activity. As you know, pepsin activity is very active in acidic condition, that is, which is pH 2. Okay, while for experiment for B, so okay, for B solution in test tube B, the solution become cloudy. Okay, because there are no reaction occur. There are no reaction occur because in solution, this solution, absence of pepsin. Okay, so there are no pepsin present to break down albumin. Okay. So as a conclusion, okay, conclusion for this experiment from your textbook, pepsin hydrolyzed protein in the albumin suspension into polypeptide. So yes, hypothesis is accepted. Okay, so albumin break down into polypeptide with the presence of enzyme pepsin. Okay, so that is conclusion for this experiment. Now we go to the last experiment, last activity. That is activity 9.2. Okay, studying the digestion of lipid in food sample. So background of this experiment is about digestion of lipid that occur in duodenum. Okay, near the duodenum, important organ near the duodenum is pancreas. Okay, pancreas, it secrete pancreatic juice. Okay, it produce and secrete pancreatic juice. Pancreatic juice, it contain pancreatic amylase, which digests starch, okay, to maltose. Okay, it also contain trypsin. Okay, and it also contain enzyme lipase. Okay, then near the duodenum also you can see gallbladder. Okay, this is gallbladder. Gallbladder, the function of gallbladder, it store bile that synthesis by liver. So function of bile is to emulsify lipid. Okay, in duodenum, it emulsify lipid into tiny droplet of oil. Cikgu Mio dah cerita tadi, apakah fungsi bile di dalam duodenum. So I'm only focus, okay, reaction between lipid with enzyme lipase to produce fatty acid and glycerol. So let's watch in this video, okay, to see how to conduct this experiment.
Okay, now we move to the activity 9.3 to study the digestion of lipid in a food sample. Let's start our experiment.
As always, before conducting an experiment, first list some of the following. The problem statement. What is the effect of lipase on lipid? Hypothesis. Lipase hydrolyze lipid into fatty acids and glycerol. For variables, manipulated variables, the presence of lipase, responding variable, time taken for the phenolphthalein indicator to turn from pink to colorless, and fixed variable, temperature at 37 degrees Celsius, volume of cooking oil, and combined volume. Make sure the materials and apparatus used are correct and everything is on your table. You also need to read the experiment procedure and run the experiment step by step. Use the diagram as a guide for you to organize the material and apparatus. Good luck, class.
Okay, before I continue the video, so I want to tell you that this experiment 9.3 is a new experiment in syllabus KSSM. Before tak ada eksperimen ini, eksperimen kalau study tentang lipid pun dia hanya menggunakan apa nama tu kertas turas. So this is new experiment. Okay, so please watching this video how to conduct this experiment.
change the color from pink to colorless is 30 seconds, while for the test tube B, the color remain unchanged. Thanks for watching and see you soon. Bye.
Okay, so for discussion, discussion for this experiment, as can you see, in a solution in test tube A, the color of phenolphthalein, phenolphthalein, change to colorless or color, it follow color of dishwashing liquid. Okay, untuk eksperimen ni, warna phenolphthalein dia mengikut perubahan warna dia. Kalau dia berubah menjadi colorless, so dia akan berubah mengikut warna bahan pencuci yang kita gunakan. So for the experiment, our dishwashing liquid, the color is green. So that's why warna tu dia tak bertukar colorless tapi dia berwarna hijau. Okay, ikut warna asal kepada pencuci yang kita gunakan. Okay, so if you can see, okay, why the color change? Why the color change? Actually, phenolphthalein ni dia sebagai indicator. Okay, it used as indicator to show that alkaline solution about pH 10. Kalau dalam persekitaran pH 10, so phenolphthalein ni dia akan berubah warna kepada warna pink. Okay, kalau pH 10. Untuk test tube A ni, solution dalam test tube A, the color turn colorless ataupun back to color of dishwash. Okay, because digestion of oil occur in test tube A. Okay, digestion of oil by enzyme lipase, it produce fatty acid and glycerol. So fatty acid and glycerol, okay, when it produce, it will neutralize the alkaline solution here. Okay, so penghasilan fatty acid dan juga glycerol ni dia akan meneutralkan ataupun merendahkan pH, okay, pH larutan yang alkali tadi. Okay, so tadi kita nampak kan dalam eksperimen tu Cikgu Izzati dia campurkan dengan pencuci, bahan pencuci. Okay, dishwashing. Kenapa dia letakkan bahan pencuci? So tujuannya adalah to emulsify oil. Okay, to emulsify oil to droplet of oil. Tujuannya adalah untuk mengemulsifikasikan minyak tadi kepada titisan minyak yang lebih kecil. So dishwash ni dia berfungsi macam bile, okay, macam bile in your duodenum. Okay, for solution B, pink color remain same because there are no present of enzyme lipase. So oil dalam ni dia it not break down to fatty acid and glycerol. So that's why the color is remain pink. Okay. Okay, as a conclusion, okay, if you read the hypothesis in activity 9.3, lipase hydrolyze lipid into fatty acid and glycerol, yes, hypothesis is accepted. Okay, as a conclusion, hypothesis is accepted. So lipase break down tiny droplet of cooking oil in this experiment to fatty acid and glycerol in alkaline condition. Okay. So habis eksperimen, ketiga-tiga eksperimen ini. So mungkin ada pertanyaan di kalangan pelajar. Bertanya kan, mungkin ada pelajar tanya, kenapa kita gunakan kalangan pelajar break down tiny droplet of cooking oil in this experiment to fatty acid and glycerol in alkaline eksperimen ini. So mungkin ada pertanyaan di kalangan dalam eksperimen tadi kita ada gunakan sodium carbonate solution. Sodium carbonate solution tujuannya adalah untuk menyediakan persekitaran beralkali, okay, untuk larutan tersebut bagi aktiviti enzim lipase. Okay, so sebagai penamat untuk sesi cikgu pada pagi ini, so let's playing a game. Okay, mari kita bermain satu game. Okay, kejap ya, tadi ada. Kalau kamu boleh nampak dekat skrin tersebut, sila type perkataan join myquiz.com dekat Google. Okey, dekat Google and enter join code. Kemudian tulis nama kamu dan nama MRSM. Okey, tulis nama kamu di hujung belakang nama kamu. Kamu letaklah nama MRSM kamu. Okey. So let's play the game. So cikgu bagi masa 2 minit. Okey, bagi siapa-siapa ni. Okey, nama akan terpapar dekat ni. So cikgu akan bagi masa lebih kurang 1 minit untuk tunggu. Okey, Chan William daripada MRSM mana? So boleh letak nama MRSM dekat belakang. So kita tengok MRSM mana yang hebat pada hari ini. Nadira daripada MRSM KB. Okey. Alia MRSM Pendang, Anis MRSM PC, Syaira MRSM LGK. Apa LGK ya? Okey, Irfan MRSM ku. Mana MRSM Betung ni? Tak masuk ke MRSM Betung? Okey, sila masuk lagi. MRSM Betul tak ada ke? Mukah ada wakil daripada Mukah. Langkawi ada, Muar ada, Izzati MRSM. Ingat cikgu Izzati tadi. Okey, guna nama pendek ni cikgu tak mesti nama penuh MRSM Okey, mana MRSM Betul? Tak ada MRSM Betul. Okey, sila join lagi. Kita akan start sekejap lagi bila ramai-ramai dah join. Kita tengok top player daripada wakil daripada MRSM Betung, Tan Yiji. Pada hari Arah tanya 517487. Sorry. Okey, ada kod dekat atas tu. Okey. Baru nampak, cikgu dimaklumkan kita ada 900 lebih kehadiran hari ini. Okey.
50 je.
Itu soal ada cikgu. Okey, merancung
Ini betul ya cikgu.
you are got alamak. Okey. Merancung pergi Betung, pindah terus ke Betung pun tak ada hal. Okey, pindah Betung tak ada hal. Okey, dialog okey. Boleh mula Now, 8 soalan sahaja. So okey. Diam, saya tunggu. Dah mula kan? Quizy. Okey. MPC, pangkalan cepa kan? Okey, tahniah cikgu ucapkan kepada Ishraf daripada MRSM MPC. Ke ada lagi yang belum habis bermain? Okey. Aiman daripada Felda, tempat kedua. Okey, 6,840 markah. Okey, top Ishraf daripada MRSM MPC. Presiden 5C, Presiden kelas 5C daripada MRSM MPC. Okey, nombor 3. Tak tahu siapa nama dia, mendapat markah 6,650 markah. Okey, so rasanya semua dah jawab. Okey, so inilah ranking keputusan. Boleh jadi ianya keluar dalam paper 1 dan boleh juga ianya keluar dalam paper 2. So that's why bila kita buat eksperimen tu, part paling penting adalah part perbincangan. Okey, bahagian perbincangan supaya kita mendapat sesuatu daripada eksperimen tersebut. Okey. Okey, tak ada lagi ya. So itu sahaja daripada cikgu pada pagi ini. Cun pukul 11.58 dan kita akan tamat tepat pada pukul 12 tengah hari. Okey, cikgu serah balik kepada cikgu Tanti. Mana cikgu Tanti, tamatkan terus ke?
Saya rasa kita sudahkan program kita dengan kita kita sudahkan dengan tasbih kafarah dan surah Al-As.
Okey.
Okay, all the best to all students.
Okay, good luck
We
people.
are great and thankful to all of you. InsyaAllah A+ untuk semua biologi.
InsyaAllah.
Amin ya rabbal alamin. Semua moderator, semua fesi dan cikgu-cikgu dekat sini mendoakan anda untuk mendapat A+ biologi.
Okay.